Okay, we're gonna go ahead and start. I hope everybody can hear me. I know you can. So, let's see. We're going a couple things on. We got the first test opens up on Thursday morning at 12:01, and we'll have. For those of you that are taking it in class, we can have it in here. You don't have to take it in here. You can take it online, but if you want to come to class and take it, that's fine too. You have the option. So you can show up then on Thursday, and that's all we're going to do is do the test. So I'm going to talk a lot today to make up for that. So lab this afternoon is going to be looking at the axial skeleton. You should have gotten an email from me last night on two good study areas for the bone test. One is an online, well, actually it's a PowerPoint review that lets you go through. It's like 44 images, and you have look at it, and the next slide tells you what it is, and that's really good. And then the other one is one that you can download and make fill in the blanks. You can identify, you can go over it and practice it over and over and over again. Both of those are very good, because the key to success on the bone test is memorization. It's how do you got to remember everything. You have to be able to memorize and to learn these bones. And I'll tell you, once you learn them, it's pretty hard to forget them. And it's going to be something useful to you later on in whatever further program you go into. It is, but the key is to practice it over and over again in various ways. Teach it to yourself, teach it to each other, teach it to your study group, quiz yourself on these blank sheets. Quiz yourself on this PowerPoint program that I've sent you. So that's the best advice I can give you to learn these bones. Yes, it's tedious, but you do a little bit at a time, so it isn't too tedious. Because if you try and do it all at once, you'll just get overwhelmed. Yes, ma'am.
Question about the lecture test.
Yes.
Is it mainly going to be like definitions and stuff, or is it going to be images as well that we have to label?
There are no images on the lecture test. It's all multiple choice.
Okay.
So it will focus on chapter 1 and chapter 2. And I tried really, really hard to keep it divided up so that there isn't a preponderance of chemistry in there, because that would have been really easy to load up the chemistry questions in that test. But I'm trying to give equal weight to chapter 1 also, as even though it's only introductory material, it should be fairly simplistic. So, but yeah, it's all multiple choice for you. And it should, if you take it online, it shouldn't take more than 40 minutes. I will tell you that even though there is a timer running online, and those of you that are in the Zoom class that are going to take it online, even though there's a timer running, the timer is not going to shut you down. Okay, it's just there so that you can keep track of how long you've spent, but there is no, you're not going to be timed on the test. Now, if you run for 4 hours in the test and get a perfect score, that may raise a red flag somewhere. But most people will complete this test in about 40 minutes or less. It's only 50 questions. So, and the tests, all the online tests are going to be different. They're all drawn from a test bank. And so everybody gets a different test in different order with different questions. And I haven't decided what part of that test bank I'm going to draw yet for the lecture test in here. So there'll be some subset of the test bank for you all.
When does the online test close?
The 24th, 5th, 6th, somewhere in that time frame. I'm giving the online, giving them a little more flexibility on timing. I mean, you have the same option, so trying to avoid any kind of conflict with anybody's schedules on this. So, because I know how things happen, I know how life gets in the way of planning for a test, or you're just not quite ready for it. So we've all been there. Let me. Anybody have that dream yet where you're going to school and it's the last day of class and you've forgotten to go to class for the whole semester, or you've had that, yeah, or you show up and it's a test you're not ready for? That's a very common dream. We all have that kind of dream. So if you're having that now, it means I'm stressing you. No, it means that that you're it's welcome to the club. I'll just say that. For that effect. So, and we'll talk about dreams when we get into the nervous system and the different types of dreams we have. And that is a very, very common dream. I mean, I still have them, and I'm on the other side of the desk with the test. But I still dream. I can't get to the classroom, or whatever. So, okay. Let's go. Any other questions? From anybody on the test coming up?
Is our study guide kind of like a test? Like the stuff that's on the study guide is what we should be studying? We should go over all the notes we took. You should go over
the PowerPoints are where I've drawn the test from, but the study guides are very useful in the same topic areas. The practice test is similar, not the same questions, but it's a similar type of topic. And there's only so many ways to ask the same question. So. I would sort of combine, go through all three. Don't fixate just on the study guide because you might miss something. Look at the PowerPoints. If there's something in the PowerPoints I didn't mention, don't worry. Then I didn't ask a test question on it. It's the only material that I've mentioned is fair game. Yeah.
Where is the practice test?
They are in assessments, and you go into eLearn, go into assessments, and go into quizzes. And they pop up there. Okay. And the first test then is right now it's down at the bottom because it hasn't turned on yet, but it will probably go right to the top when it opens up. So you can take the practice test. Practice test you can take as many times as you'd like. If you've got nothing else to do and you're really bored, and you can take it a couple times. So okay. Any other questions on that? Okay, well, I'm going to go ahead and get into back to cells here now. And let's see, if I remember correctly, we had stopped on fluids and isotonic solutions and stuff like that. And I think we're ready to talk about active transport. So let me go into share screen. And bring this up. Okay. We've been up until this point, we've been talking about passive transport. And passive transport, cells love passive transport. Passive transport doesn't cost the cell anything. It's a free ride. Gases are a free ride. Waters are a free ride, even though they have to use a special channel. Certain ions are a free ride. They come in and out of the cell all the time. The cells love that. When things go down the concentration gradient through diffusion or osmosis, or even facilitated diffusion, they love that because there's no cost involved. But sometimes cells have to move stuff across their membrane, but it's coming against the concentration gradient. It's going in the opposite direction. It's going essentially uphill to go into the cell or out of the cell. They have to work harder. You may have a low concentration of something outside, and you need to bring it into the cell where it's high. You may have an ion outside the cell that you need. Lots of ions are inside the cell. It's not going to come in passively. It's not going to diffuse because the high concentration is inside. It's busy trying to leak out. You got low concentrations outside. You want to get them, and so you have to pump them in. Sometimes you have low concentrations of something inside the cell, and you want to get it back out. Well, it's, things don't diffuse from low to high. We have to pump it from low to high. It's like pumping uphill. And this is what we call active transport. Active transport moves against the concentration gradient. It's like when you're at Walmart and you're bringing your buggy down an aisle, you got 500 people coming at you the wrong way, and you're trying to go the wrong way to you. You're moving against the flow. That's you got to spend more energy to try and push your buggy up against that. That's active transport. Now, cells use active transport to the extent that about 40% of the amount of energy that a cell uses is simply on active transport. So it's burning up a lot of energy here. It's burning up a lot of glucose to pump substances across its membrane. Now, there's two ways they do this. They have what are known as antiports and symports. An antiport is what a cell does when it pumps one thing out of the cell and a different substance into the cell at the same time. It uses the same mechanism. You're pumping sodium out of the cell while you're bringing potassium in, for example. That's a real example. Or you're bringing two things in together. Lots of times cells will bring sodium and glucose, strange combination, but sodium and glucose will come in together across the membrane. And this process always is going to require ATP. Now, the key here is for active transport, we talk about primary active transport, and this is what we use ATP for. This is the actual pump. There is a pump embedded in the cell membrane. There are a variety of pumps. There's a hydrogen pump, there's a calcium pump, there's a sodium and potassium pump. A good example of hydrogen pumps, or what we call proton pumps, are what we have in our stomach. Proton pumps are pumping hydrogens out into our stomach. That's what gives us acid reflux. If you take famotidine, or you take Pepsid, or anything like that for reflux, or as we say, heartburn and indigestion, what you're doing there is trying to shut down a proton pump embedded in the membrane. Now, the most important pump that we deal with is called the sodium and potassium pump. Sodium-potassium pump. And what happens here in these pumps, and all these pumps are involved in what we call primary active transport, is that this pump embedded in the membrane is going to open up a carrier protein and allow one ion to go out at the same time another ion goes in. And the reason you use energy is that you're going against the concentration gradient in both directions. Now that's primary active transport. Secondary active transport doesn't cost any energy. It's taking advantage of the fact that you've pumped sodium out. You've pumped sodium out, and guess what it's going to do? It's going to leak back in, because there's lots and lots of sodiums on the outside of your cell. That's what cells, that's the number one ion outside your cell is sodium. So the more sodium you pump out, the more it's going to leak back. And when it leaks back, it usually drags along a glucose. I don't know why glucose and sodium are so friendly, but a lot of times that's what happens. Sodium will enable glucose to come back in. The reason sodium can leak back in is because we keep a high concentration outside thanks to our pump. We call it secondary active transport, but we're not really using any energy, any extra energy to get for the sodium to come back in. Anyway, now, active transport. We got all sorts of channels in our cell membranes. This active transport with sodium and potassium, we know more about the sodium potassium pump than any other pump out there. It's only been recently when we discovered about the activity of proton pumps in the past 20 years or so, and the whole concept of acid reflux. that these proton pumps have become more, been identified. The sodium potassium pump will pump three, this is an important thing to remember, 3 sodiums out of the cell. 3 sodiums out of the cell and 2 potassiums back in. See, our cell membranes, I wonder if this, no. Our cell membranes are always in our bodies. Come on. There we go. Sure. Our cell membranes on most of our cells, and this is a cell membrane right here. It's a highly detailed drawing of a cell membrane. Most of our cells have lots of sodium ions floating around here outside the cell in the interstitial fluid. It's mostly sodium on the outside of the cell. Now, the number 1 ion inside the cell is potassium. We have potassium ions on the inside. There's more sodium outside than there is inside. So what's sodium going to do? Sodium's going to be leaking into the cell. Sodium always leaks in. That's what it does. Because it's, high sodium here. That wasn't good. Low sodium in here. That says low. So sodium's always leaking into the cell. We don't want sodium in the cell because that interferes with the membrane. There's a charge on the membrane here. There's a charge on all of our cell membranes, and the charge is based on the difference between the sodiums and the potassiums. Well, sodium's always leaking in. If enough sodium leaks into the cell loses its charge and can't do anything. Because our ability to contract our muscles and send nerve impulses depends on these electrical charges on our cells. And the only reason we have the charge is because of the difference between the sodiums and the potassiums. It's like a battery. The outside of the cell is the positive side, the inside is the negative side. If the sodiums leak in, the cell becomes neutral and it can't do any work. So sodium's always leaking into the cell and we're always pumping it back out. At the same time, we have high potassium on the inside and it's leaking to the outside. So what the sodium potassium pump does is it sends sodiums back out and recovers the potassiums. And this is what it sort of looks like. It opens and shuts. Sort of looks a little like a Pac-Man. I just noticed that. So you can see it's kicking 3 sodiums out every time and bringing those 2, the potassiums are green. I don't know why they're green, but the potassiums are green, the sodiums are yellow. And every time the pump works and kicks 3 sodiums out, it takes ATP, but it also opens up to bring potassiums back in. And we're trying to keep sodium's high on the outside of the cell and potassium's high on the inside, so we keep a charge across the cell membrane. In muscle cells, it's minus 90 millivolts. It's a 90 millivolt charge on the surface of the cell membrane. That's the potential, the electrical potential of the cell. That means that cell can contract muscles. It can, nerve cells have a charge of minus 70. They can send nerve impulses, but only if we have high sodiums outside and high potassiums inside. And the thing to remember, the one thing to remember about the sodium potassium pump is this phrase right here: 3 sodiums out of the cell and 2 potassiums back into the cell. So 3 sodiums out, 2 potassiums in. 3 3 sodiums out, 2 potassiums back in, because we want to keep high sodium here and high potassium here. We don't want to have sodium on the inside. We don't want to have potassium on the outside. So that's what we're trying to do. Now, okay. So that's the sodium potassium pump. And it is a symport. I'm sorry, it's an antiport because sodium goes out, potassium comes back in. If you have two molecules going in opposite directions, it's an antiport. Now we also see an example here of a symport, because there's our sodium potassium pump on the left there, kicking it, sodium's out and bringing potassiums back in. But a symport usually shows up in secondary active transport. For whatever reason, when sodium lands on an ion channel that will also hold glucose, when sodium lands on the ion channel, when sodium lands on this ion channel here, this channel changes its shape. Sodium comes in, lands here. This causes this structure here to change its shape for glucose. And that means this glucose molecule right here can come and land in here. Now that channel won't open up to the inside of the cell until glucose lands on it. But as soon as it does, it changes its shape. Sodium comes back into the cell. Glucose comes back into the cell. And we get more glucose going into the cell. Now, does this is this working, in spite of insulin? Well, insulin only works when, to generate carrier proteins when we've got high blood sugar levels. What if there's just normal glucose, normal sugar out there? The cell's bringing more sugar in. Maybe we're not running at excessively high levels of sugar, but we still have more sugar that wants to come in. We have more sodium. Is sodium maybe piggybacking the sugar, sugar with it? So just something to keep in mind. Secondary active transport doesn't use any additional ATP, but it's another way to get sodiums and glucoses into the cell. Now, the other way that cells get things moving that takes energy is what we call vesicular transport, or transport using bubbles called vesicles. Now, a cell has two mechanisms: endocytosis or exocytosis. Endocytosis is moving something into the cell. Endo, think of entrance. Endocytosis. Have you ever seen an amoeba move around, and a picture of an amoeba doing something, engulfing a bacteria? That's endocytosis. It's actively chasing down something and eating it. Up here we see white cells in the top there engulfing a bacteria. Bless you. So that is active. This is transport using a vesicle, because when the white cell engulfs the bacteria, it puts it inside a bubble called a vesicle. The vesicle has the same membrane as the cell membrane. It engulfs it in there, and then it proceeds to digest it and destroy it. It could also be a fragment of a neighboring cell, because when cells get old, they start breaking apart, and all their friends and neighbors come over and eat them. It's a very tough homeowners association. You don't get fined, you just get eaten. So, but because cells will only stay alive for so long, and then they start to break down. They will start releasing enzymes, digestive enzymes, to break themselves apart, but then their friends and neighbors will step in and finish the job. Barbecue at Fred's. But that is endocytosis. Exocytosis is when a cell is making something like a protein. you're busy making proteins. You're busy making hemoglobin. Now you have to get the hemoglobin out of the cell. So you put the hemoglobin protein in a vesicle and you fuse that with the cell membrane and you get to release it. Like we're seeing here in the bottom on the animation there in the bottom. The vesicle comes up, fuses with the membrane. You never open up the inside of the cell. You just open up the vesicle and the contents get released. So endocytosis is going into the cell. Exocytosis is leaving the cell. Now, endocytosis gives us three categories. We have what's known as phagocytosis, which is cell eating. You know, the amoeba eating a bacteria, a white cell eating a cell fragment. This is phagocytosis. We have very large white blood cells called macrophages, which means large cell that eats things, essentially, and they go around and they kill bacteria and they destroy viruses and they destroy infected cells, and that's what they do, and it's cell eating. Pinocytosis is literally cell drinking. Fluids that the cell wants, it can go and engulf those fluids and bring it into the cell. And so we see phagocytosis, cell eating, pinocytosis, cell drinking, and then receptor-mediated endocytosis is when there are certain things that we want to bring into the cell that we only have very specialized receptor sites for. So, for example, here you see on the far left-hand side, you see the plasma membrane engulfing a food particle. It forms a vesicle, or they call it a vacuole, but it's a vesicle. It's the same cell membrane structure around the vesicle. It just pinches off, and then it brings it into the cell, and we let the digestive enzymes break it apart. The same thing happens with the vesicle for fluids in the middle. And the third example is where we have the specialized receptor for some molecule that we want, and only that molecule gets picked up there. But all three of these require energy. Now, exocytosis, we'll be producing proteins and store them in a vesicle that will move to the surface of the cell, fuse with the cell membrane. You'll notice that it never tears a hole in the membrane itself. It just fuses with it, and it opens up the vesicle so whatever the protein can leave, for example, and we release it into the fluid around the cell. That's what we do with proteins and hormones and things like that. We release it into the fluid, where eventually it ends up in the plasma and gets distributed to wherever it's supposed to go. And this is really what it looks like. This is about 100,000 magnifications of some vesicle releasing something for us. So, and again, this requires energy. Okay, so that's leading up to the material inside the cell membrane. We're talking now about the cytoplasm and the organelles within the cytoplasm. Now, the cytoplasm is the goop inside the cell. It is sort of a watery mixture. Some say it's like a gel. Others say it's more water-based. It contains proteins and salts and sugars, and it has organelles, which are little structures within the cytoplasm that do things for the cell, like break down sugar for energy, or produce proteins, or destroy free radicals, or fight bacteria. We have, so all these structures here are organelles. The largest organelle, of course, is the nucleus, and then we have, and every one of these structures on the left-hand side, like the mitochondria, has a membrane around it, just like the nucleus does. The nucleus has a nuclear membrane made of the same phospholipid bilayer as the cell membrane. So do all these other organelles on the left. On the right-hand side, those structures are inside the cytoplasm, but they don't have that membrane. So now let's talk about the mitochondria. You'll hear more about the mitochondria in chapter 24, which is actually the chapter 4, but we still call it chapter, never mind. Okay, the mitochondria. Most textbooks call this the power plant of the cell. Better term for it is the energy conversion center, because what they're doing is mitochondria is where sugars are essentially oxidized, they're burnt, if you will, to release their stored energy. And that stored energy then is used to make ATP. Now the glucose, the sugars contain lots of energy. And we will take that energy, we can't take it directly to the cell. We can't release energy from the sugar. We have to park it in ATP and transport it where it's needed using that ATP molecule we talked about the other day. But each ATP molecule can only hold about 7 kilocalories of energy, and it only lasts for about 5 seconds. So we're always converting the potential energy of sugar into kinetic energy and then back to potential energy in the ATP molecules. So the mitochondria is really where we convert the chemical energy from one form of chemical energy to another form of chemical energy. Very important structure for us. Without mitochondria, we couldn't survive. Okay. So, and cells that are going to be very, very busy and very active, like muscle cells, are going to have lots of mitochondria because we need lots of energy for our skeletal muscles. Now, something else we find, this is one of the, this is like the top organelle without a membrane. These are called the ribosomes. The ribosomes are where we put our proteins together. That's something else that's very important for a cell to do. The DNA controls everything a cell is going to function with, all cell functions. If we need to make a new protein, let's say we need more hair color, eye color, skin color, hemoglobin, various enzymes, whatever protein we need, collagen, whatever, elastin, we will send instructions out to the ribosomes in the cytoplasm on how to assemble the amino acids in the right order. And it's the assembly of proteins takes place at the ribosomes. If we couldn't assemble our proteins at the ribosomes, our cells would cease to function. And then shortly thereafter, we'd cease to function. 2 types of ribosomes. Most of them are bound to what we call the endoplasmic reticulum, series of tubes that surround the nucleus, and it's studded with millions of ribosomes, and so many that we call it the rough endoplasmic reticulum because it has all these little bumps on there. And this is where we make most of our proteins. Free ribosomes are present in other areas in the cytoplasm. We tend to see those making things like antibodies, maybe soluble proteins in here, but most of our ribosomes are going to be are attached to the endoplasmic reticulum. Now, exactly what is this endoplasmic reticulum and why is it so hard to say? It's a series of flattened tubes that completely surround the nucleus. There's no good illustration that shows this. But there's our nucleus. The nucleus is the ball in the middle of the cell, and surrounding that are is a larger ball containing all of these tubes. And the endoplasmic reticulum and the nucleus share common openings. There are what we call nuclear pores. Those are openings in the membrane of the nucleus that open directly into the rough ER. We call it the rough ER because that's where all those little dots on there, those are the ribosomes, the little tiny dots of the ribosomes. When messenger RNA is assembled inside the nucleus. Messenger RNA is a copy of the gene, because remember DNA, the structures of the DNA, it's all these nucleotides linked together in a specific order, and a gene is nothing more than the physical location for a set of instructions on the DNA, like hair color here, eye color here, skin color here, hemoglobin here. It's just physical location. When we need to make some more protein, we make a copy of those instructions. And we use messenger RNA, the single strand version of DNA, to take that out to the ribosome. And the way it gets out is to go through those openings on the nuclear membrane out into the endoplasmic reticulum. That's all it does. Now, the other type of endoplasmic reticulum, or ER, is the smooth ER, and it's a much more fascinating structure. Smooth ER has no ribosomes. That's why it's called smooth. Clever name there for that. So rough ER is where we make proteins. We make our proteins and our phospholipids all in the rough ER. We assemble them at the ribosomes. The smooth ER, which is still continuous with the rough ER, does a whole lot more. We break down lipids in the smooth ER. We make steroid-based hormones in the smooth ER. We make the fats, the lipids for the lipoproteins. We transport fats in the smooth ER. And this is a big one. The detoxification of certain chemicals. Remember that everything we eat and everything we drink ends up in our plasma. Before it can be circulated through our bodies, though, when we absorb it at our small intestine, the first place it goes, everything we eat and drink goes to the liver. Our liver has a vast network of smooth endoplasmic reticulum to detoxify the food additives and the chemicals and the dyes and the preservatives that show up in our food. Pharmaceuticals are broken down to a safe dose for us. For example, Tylenol, we all know what Tylenol is, right? Painkiller. Average adult dose is 1,000 milligrams to 500 milligram tablets or capsules or whatever. Before that can go into our body and function as a painkiller, it has to go through the liver. So the liver will break down a fair amount of that Tylenol. And whatever comes out of the liver is what we get to use to kill pain, to block. Tylenol works by blocking pain signals. That's all. That's what it does. We don't get the full 1,000 milligrams, so our cells can't process that much, can't deal with that much. The liver knocks it down to a level where it's not going to hurt us. And all of our medications are based on the premise or the knowledge of what the liver will do to certain drugs. That's why we have adult doses and children's dosages. You wouldn't give 1,000 milligrams to a 5-year-old. You may not give 1,000 milligrams to an 85-year-old. The 5-year-old's liver can't deal with the adult dose, nor can its body. The 85-year-old may have a liver that's not, what's an 85-year-old liver, and it may not be up to the task of dealing with 1,000 milligrams of Tylenol. Maybe the elderly patient has to have a smaller dose of this because the liver processes all these chemicals and tries to break them down and destroy what are the harmful chemicals. I mean, Tylenol is considered everything is considered a harmful chemical. A toxicologist will tell you that everything's a poison, and they'll tell you that the dose makes the poison, because anything can kill you. Water can kill you, salt can kill you, oxygen can kill you. It just depends on how much it is. The liver is going to break down these chemicals for us, to try and render them harmless. Alcohol, for example, alcohol is a good example. If you drink alcohol, if you know somebody you know, the classic, well, I've got this friend that drinks alcohol, you know, so that what the liver does to alcohol is it starts breaking it down. The end result is it is converted to carbon dioxide and water. Most things that we eat and drink end up as carbon dioxide and water. The liver can process an ounce of alcohol per hour. That's like one bottle of beer, one mixed drink, a small glass of wine. You know, if you were to drink a bottle of beer and make it last for an hour, and then drink another one and make it last for an hour, you would have no ill feelings the next day. You would have no consequences of you know, no hangover, no dehydration, no nothing, because you allowed your liver time to process the chemical, process the alcohol. But we all know that drinking 1 ounce of alcohol over the course of an hour is probably not what most people do. You know, it's beer is going to get warm and flat, you know. So most people will drink a little more than 1 ounce an hour. I mean, and what happens is the liver can't keep up. And the consequences are that if you have a patient that shows up in the emergency room and they've indulged heavily in alcohol, depending on how much they've taken in, they can be suffering from alcohol poisoning, and they can die from that. You know, excessive consumption can kill your patient. More than likely, your patient's going to be have nausea and vomiting, and probably have a bad hangover the next day, you know, simply because they overindulged. Now, the hangover is caused by the breakdown of the alcohol in the liver. The liver can only do so much. As the liver breaks down alcohol, the intermediate steps include converting that alcohol to formaldehyde, which is also known as embalming fluid. And normally what happens is that formaldehyde is formed and then broken apart instantly. So, you know, if you're going the drink an hour, you don't notice it, because it's like as quickly as the formaldehyde forms, it's gone. And something called methanol, which is a toxic version of alcohol, it's called wood alcohol, and that also forms and then disappears. But if the liver gets backed up, this accumulates. And so over the course of the next day, your patient is suffering from a bad hangover because the liver is still trying to get caught up. And of course, on top of all that, your patient's likely dehydrated, because one of the things alcohol does is it interferes with our ability to regulate the need to urinate. Alcohol suppresses the control mechanism in our brain that says don't pee. And so alcohol says you want to pee all the time. And when you do, you get dehydrated. And one of the other consequences of this hangover is severe dehydration. So all because the liver is trying to do its job. Anyway, now, other things that the smooth ER does for us, when we have glycogenolysis, the lysis of glycogen to glucose that takes place in the smooth endoplasmic reticulum. And when we get into muscles, you're going to see that we store lots and lots of calcium in a type of smooth ER around the muscle cells, because calcium is absolutely essential to a skeletal muscle being able to contract. So we store lots of calcium in there. So the smooth ER is a whole lot busier than the rough ER is. Smooth ER has a lot of functions in there. Okay, now, other structures. The Golgi apparatus. The Golgi apparatus is the FedEx of the cell. When we make proteins in the rough ER, the proteins are all collected together in a vesicle and shot out of the rough ER, and you've got hemoglobin, eye color, hair color, skin color, enzyme this, enzyme that, all in this vesicle, all mixed together. They get sent to the Golgi apparatus. And the Golgi sorts it all out. And at the end of the Golgi, you get a vesicle containing nothing but hemoglobin, nothing but hair color, nothing but eye color. It's like FedEx at their big sorting facilities. You know, everything going to Chicago goes here, everything going to Milwaukee. I don't know why you'd want to, but it's going there, and so on. And that's what the Golgi does. And the Golgi looks like this. It sets below and the rough ER, and the vesicles are coming in. You see that blue vesicle there? I don't think it's really blue, containing all the proteins just thrown together. And then as those proteins are moving through the Golgi, those flattened sacs, they get collected so that all the same type end together in a vesicle, and then that gets released. Now there's 3 outcomes for that. Those proteins that are in there. You can release them to the outside of the cell, you can keep them in the membrane, or you can pack them off to another organelle called the lysosomes, where it's going to function to destroy things. So this is not a bad drawing here, but this is because this is really what they look like here at 90,000 magnifications. The vesicles, the transport vesicles come in here, and out come the secretory vesicles here with all the proteins. So here are the 3 choices you get. You can release them. You've made a hormone, a protein-based hormone. You release the hormone, goes out into the interstitial fluid, and then ends up in the plasma and goes to wherever it's going to go. Or it's going to be a channel protein, a whole bunch of channel proteins. Maybe this is how we get the channel proteins and carrier proteins for insulin. Insulin tells the cell to make lots of carrier proteins for glucose, and so those carrier proteins end up in the cell membrane. Carrier proteins are proteins. They got to get made in the rough ER. Insulin triggers that. So we make these carrier proteins and glucose can come in. So we stay in the membrane. The third choice is to end up, the vesicle contains lots of digestive enzymes, and it becomes a structure known as a lysosome. And this is what the cell uses to break down, worn out organelles or worn out cells, or to go out and kill a bacteria, if you will. Peroxisomes uses hydrogen peroxide. Peroxisomes destroy free radicals. A free radical is a molecule or a fragment of a molecule that forms, usually contains the wrong kind of charge on it. And as it travels through tissues, it destroys healthy tissue. Free radicals are likely a cause of cells becoming cancer cells. Imagine a hydrogen ion. When we think of a proton, it has a positive charge. But think of a hydrogen ion with a negative charge, or a glucose molecule with a charge on it at all. It's not supposed to have a charge. Peroxisomes work by breaking down these radicals, what we call the free radicals, using oxidase, and that converts them to hydrogen peroxide. Now, this isn't the same stuff while it's the same formula that, you get down to Walgreens and get one of them brown bottles, and it's, H2O2, hydrogen peroxide. It's an antiseptic. Pour it on a cut, it bubbles and foams, and it doesn't hurt, and it's all good. Well, same molecular formula, this is much more highly concentrated. It's toxic to the cells. So after we make the hydrogen peroxide, then we break it down using something called catalase, and we end up with water. Oxygen and water get released. So we use this process to get rid of all these free radicals, which abound in there. We got to get rid of these free radicals before they destroy and damage healthy tissues. Okay, now, lysosomes. I've been talking about lysosomes here. Lysosomes are digestive enzymes. They are so dangerous to the cell that they have to be contained within a vesicle that resists their activity. because they would dissolve away the cell. If the lysosome were to open up, and it can open up, the cell would be destroyed. In fact, when the cell decides it's time to go and initiates apoptosis, which is cell self-destruction, it will release its own lysosomes and dissolve itself. Lysosomes are what we use to, when a cell engulfs food, it will then fuse the vesicle with the food in it. With a lysosome, it will dissolve it and release its nutrients. It's what it kills a bacteria with. It's what a white cell uses to destroy a virus. It's what it will use to destroy a damaged cell. These are, it's like containing very strong acids, these digestive enzymes inside this little bubble. And so we also use these lysosomes for two other things. When we need calcium, there are specialized bone cells that are packed full of lysosomes that will dissolve the bone to release the calcium. We're not aware of it. It happens all the time. We're always readjusting our skeleton. We also break down glycogen with these lysosomes. So it's a very important structure, but they're dangerous. We have to keep them isolated inside their vesicles. And there is a condition, and there's a reason I bring this condition up. It's called TaySachs disease. There's a condition occurs when we don't have the right lysosome present. This is one of those recessive disorders, genetically controlled, that shows up in certain population subgroups. TaySachs occurs when the lysosome that would break down what we call glycolipids, a glycolipid is a fat with a carbohydrate chain attached to it. It's a sugar-coated fat, if you will. As we develop, as we go through infancy, as our brain's developing, we form these glycolipids because we need fat and glucose for energy, and these glycolipids will form and they will accumulate. We keep them in check with our lysosomes here. There are specific lysosomes just to hold them in check. If we are missing that particular type of lysosome, the sugary fat accumulates and starts destroying the brain and the central nervous system. And this usually results in death by age 5. There's no treatment for it. There's no cure for it. It is a genetic disorder. Now, who gets it? Well, the primary subgroup susceptible to TaySachs disease are people of Eastern European Jewish descent, because Eastern European people, the Jewish populations in Eastern Europe were pretty much a closed society. And this, let me back up. To have TaySachs, you have to have a recessive trait from the mother and the father. And in an open society, in an open genetic society where anybody can intermarry with anybody else, recessive traits like that tend to disappear over time. But in closed societies where you only marry within your population subgroup, genetic traits tend to stick around. Recessive traits tend to stick around. And Eastern European Jews were very closed. Their society was very closed. It kept these recessive traits alive in their population. And so individuals that are descendants of Eastern European Jewish people tend to have a higher occurrence of TaySachs. Now, there is another, let me go back. There is another subgroup, and this is where I tie it back into us. There is another subgroup here. It also showed up in French Canadians that lived a couple hundred years ago in Nova Scotia, in that area. And for whatever reason, they were a closed society, and they also had this trait in their gene pool. Well, if you know anything about history, the French Canadians and the English government of Canada didn't get along very well a couple hundred years ago. So the French Canadians, many of them migrated south into an area below New Orleans. And they eventually were called the Cajuns. You know, it was sort of an abbreviation or a modification of French Canadians. They were the Cajuns. Now we're getting closer to home. And as the Louisiana population spread back north and migrated, you know, what happened in Katrina? New Orleans took a big hit, and lots of people migrated north. Lots of people moved into Tennessee from carrying these same French Canadian genes. So there is a very good possibility that because of this influx of population over the past 200 years as they migrated north, that you could very easily see this type of condition. So, boy, that was a long rabbit hole I went on that one. But normally you wouldn't expect to see TaySachs around here, if at all. We tend to see it in more in the northeastern cities than we do here. But because of the influx of the French Canadian population in the 1700s, 1800s, and the migration northward over the past 200 years, those genes have been brought up here. So it's entirely possible you would see this. It's a very subtle condition, but unfortunately, you know, if it's the infant TaySachs, it's going to have a very poor prognosis. Okay. So on that cheery note, let's move on. Cells have skeletons. Cells have shapes. Why do we have nerve cells that are long and skinny? Why are white blood cells shaped like amoebas? Why do we have columnar cells or cuboidal cells? Why are muscle cells able to contract? We have skeletons. We have protein skeletons that form the shapes of these cells and allow the cells to move. You know, they can swim, they can contract. We have what we call the contractile proteins that cause the muscle cells to move over top of each other. The cells anchor themselves to other structures so they can do things. They can have shapes. We have basement membranes underneath our epithelial layers. All this is due to these cytoskeletons. Now, the skeleton has three distinct types. It has what are called microfilaments, intermediate filaments, and microtubules. We see the microtubules. We might see that in spindle fibers, for example. We might see those in cilia. We might see those in flagella. The microfilaments we see in our contractile muscle. Skeletal muscle, for example, has two types of microfilaments. One type called a thin filament called actin, and the other one a thick filament called myosin. And then we might see the third category are the intermediate filaments that are keratin, you know, waterproofing our skin, coiled together like this. So we see these various structures here, three different possibilities. So our cells can do all sorts of things, you know, thanks to these skeletons. Okay. Another structure we see in inside the cell, and I know this is one of those hard-hitting points that, you know, that just expect to be asked any time later in your career about, that was sarcasm. So I've been doing this for 18 years and no one's ever come up to me and asked me about centrosomes and centrioles. However, these little structures are shaped like barrels, and there are a pair of them. And they are found on one end of the cell. When the cell divides and undergoes mitosis and cell division, these centrioles will copy themselves, and they will be the basis of forming the spindle fibers. They will grow out from these centrioles here. And the same structure that we have forming the spindle fibers is what we see in the flagella and in our cilia. And so cilia is a little more important than that. Now, something else we see, the cell membrane forms villi and microvilli. You know, here we see, and why do we care about this? Well, if we have a cell, here's a cell surface here. This is a cell, and the cell membrane has all these folds on it like this. And then on those folds, there are microvilli that project out to the sides. That increases the surface area of the cell membrane. If you take a small intestine of an individual, 20 feet long, more or less, of simple columnar cells, if you were to take that and stretch it out, and taking into account the villi, little villi means fingers, and microvilli means tiny fingers. If you were to stretch out the entire surface area available in that 20-foot long tube, you could cover the floor of 2 classrooms. There's that much surface area, or a whole tennis court, maybe. I don't know. It's a huge amount. Microscopically thin, 1 cell layer thick, but it is this huge area for absorption by using these folds in here that stick out, these villi and these microvilli. Okay, finally getting into something better. The nucleus, the center of the cell, where our DNA is contained. Remember, every cell in our bodies that has a nucleus has a complete set of our body's DNA. It doesn't matter what kind of cell it is. If it's a cheek cell, if it's a liver cell, if it's a skin cell, if it's a white cell, the nucleus is going to have the DNA, and the DNA is identical in every cell. It doesn't matter what kind of cell it is. The DNA for that being that kind of cell is turned on. The structures in the DNA is turned on to be a white cell or a skin cell or a liver cell, and everything else is turned off. The information's there on how to be a muscle cell or a nerve cell or a gallbladder cell or a liver cell or whatever. But if you're a skin cell, you're only using the instructions on how to be a skin cell. You know, if you get a cookbook and you're learning how to, you want to make lasagna, and you got a cookbook, you're not going to be reading the section on how to bake a cake. You're going to be looking at the section on how to make lasagna. You know, the other information's there, but you're not going to use it. You're only going to use the stuff that you need. And so all of our cells, while different, all contain the exact same DNA, except red cells. Now, red cells had a nucleus. They kick it out before they're finished being made. So they're the only cells in our bodies that are enucleate or anucleate with no nucleus, and red cells don't really care. So most of our cells have 1 nucleus. Skeletal muscle cells are multinucleate, but they don't divide, so they don't care. When we are forming our muscle cells in utero, the precursors of our skeletal muscle cells are smaller cells fused together. We keep the nucleus, but we don't divide. But since muscle cells don't reproduce, we don't care. It doesn't hurt us. So we'll have multiple nuclei in skeletal muscle. We may see some bone cells, and we may see some liver cells that way, but mostly it's going to be skeletal muscle. But we can say with pretty much confidence that all cells have a nucleus in there, except for red cells. So now when you're looking at a nucleus, what do you got? Well, the outer covering of the nucleus is called the nuclear envelope or nuclear membrane. So it's the membrane of the nucleus, and it's a phospholipid bilayer, just like the cell membrane. Within the nucleus, we've got a structure called the nucleoli. That's where we're going to make the messenger RNA. And we have something else called chromatin. And chromatin consists of the DNA and a protein that the DNA is wrapped around. So here you see, here's our nucleus, there's the nuclear membrane, there is the nucleolus or nucleoli where the RNA is assembled, and these little squiggly purple things are the chromatin. That's the DNA wrapped around a protein. Within the membrane, we have holes. We have these nuclear pores I mentioned. They're just openings so that RNA can go out to the ribosome. That's all they are. The nucleolus is where we manufacture the RNA, particularly for the ribosomes. And we have a nucleoplasm, which is the equivalent of the cytoplasm in here. So let's see. So there's our nucleus. The pores are continuous with the rough ER. You can see the nuclear pores here on this first picture. All those little dots are the nuclear pores on the surface of the nucleus. There's what they look like. Those are the little openings in there. There is a network of filaments that give the nucleus its shape. That's what we call the lamina down at the bottom image there. But the main thing about the nucleus is all the DNA contained in there, the chromatin. Chromatin is 30% DNA, 60% histone, and 10% RNA. Now remember, the DNA molecules are about, each strand is about 6 feet long, and it's very fragile. So we wrap it around the histones to form, and the RNA to form this chromatin so we can protect the DNA strand. So the DNA wraps itself around the chromatin, and it looks sort of like a candy cane. With candy canes have the red and white stripes, so that's sort of what the effect we see here as they wrap around each other. Now, when it's uncondensed, the strands are all over the nucleus. You've got 23 pairs of chromosomes in every cell, and chromosomes are nothing more than the chromatin, the DNA, the histone, and the RNA. And the only time we see that chromatin is when the chromosomes only occur when they are only visible when they condense. Normally they're stretched out, and that's all a chromosome is condensed chromatin, because the DNA is very, very fragile. So we condense it into these chromosome structures. And there's what it looks like. There's when I was talking about the candy cane effect. The chromatin and the DNA and the histone wrapped around each other with this candy cane look. And when they condense into a chromosome, they're visible, because they're certainly not visible when you look at it under the microscope when they're uncondensed, because we're talking about single threads, these individual threads of DNA. You can't see it. Now, a chromosome the chromosomes contain all the DNA for a certain particular set of traits. And of course, there are multiple locations on the chromosomes. The chromosomes are paired up. We have 23 chromosomes from our mother and 23 chromosomes from our father, 46 altogether. So we have 23 pairs of chromosomes. We have a pair of chromosomes a pair of chromosome 1, a pair of chromosome 2s, pair of chromosome 3s, and so on. And they pair up and join together like this. So they, you know, when we have, when we're undergoing mitosis, we will have what are called sister chromatids linked together. Okay. Now I mentioned this a little bit in lab, and this is a term called the cell cycle. This is the life cycle of the cell. Interphase is what a cell is doing 99% of its time when the cell's busy being a cell. I'm a liver cell. Hey, I'm a liver cell. I'm doing liver cell things. Or I'm a bone cell. I'm doing bone cell things. At some point, as we approach the end of the life cycle of the cell, of a particular cell, it will replicate its DNA. If it is one of those that does reproduce, it will replicate its DNA and it will prepare to go into mitosis, and it will go very quickly into mitosis and then go into cytokinesis and form two daughter cells. So 99% of the time, I'd say even 99.99% of the time, a cell is busy in interphase, and it's a little itty bitty amount of time they are in mitosis, and then cytokinesis after that. So interphase, busy being a cell. Now there are subphases in here. G1, or gap 1. Growth, metabolism, activity, lots of busy work going on. Cells that are going to divide, many cells that divide, the bulk of their activity is taking place in the G1 phase. Now, some cells never divide, and we say they are in G sub 0. Muscle cells don't divide. Muscle cells don't replicate. Nerve cells don't replicate. You have all the muscle cells at birth that you're ever going to have. You can make them bigger, you can make them smaller, but you're not going to grow new ones. The same generally applies to nerve cells too. Nerve cells can die, muscle cells can die. You're not going to grow new muscle cells, and you're probably not growing new nerve cells too. So that's G1. The second stage here is the S stage, or synthesis stage. This is where we copy our DNA. Because if we're going to produce two daughter cells, we got to have a complete set of DNA for both daughter cells. So we got to copy our DNA. And so that's the S stage, where we copy things. And then the last stage of interphase is G2, where we're making sure we have all the organelles copied. We make sure we have all the enzymes necessary for mitosis to take place and cytokinesis. And then we slide into the mitotic stage. And the cell cycle looks something like this. Now, this is one of the better diagrams that shows you the amount of time spent in interphase, because a lot of cells a lot of diagrams will give mitosis like a whole 1/4 of the circle, and that's not true. Mitosis actually, if you were looking at it to scale, mitosis and cytokinesis would be one black line there in mitosis, like this last line right here would be the amount of time spent, because mitosis and cytokinesis is rapid. Seconds, it's over in seconds. A cell could live for 5 days and then undergo mitosis, and it'll be over in seconds, and you have two daughter cells in seconds. So interphase runs from the start at G1 all the way around to mitosis. 99.99% of the time, a cell's in interphase is doing cell stuff up until the point that it undergoes mitosis and cell division. So we have growth and DNA synthesis, and we have more growth and activity. Now, that other thing about the amount of time it takes to replicate the DNA is also very short. It doesn't take half the cell's lifetime to replicate its DNA. It's very quick because it can't live it can't survive with a whole other set of DNA floating around inside its nucleus. How could it live? How could it function with two sets of controls? So it makes its DNA very quickly before it's ready to go into mitosis. But there are a whole lot of good checks and balances in here too. Now, oh, this is a good place to stop because I got 3 minutes. I can't get into it. Okay, we'll stop here. And we will pick up with this again on Tuesday. Thursday, we'll have the lecture test, either in class or online. It's up to your choice. And lab this afternoon, be prepared to look at the axial skeleton. That's the vertical axis bones. So, and does anybody have any questions? Okay. Now we'll get us out of here, and I will see you all later.